Hello and welcome, dear students. How are you all? So let's start the class of biology. Please take your seats. Okay. So today, hello Itanagar students. How are you? Please report your attendance before we begin. Thank you. I'm fine. Alright, so Shillong, Tamenglong, Itanagar centers, please report your attendance. Alright. So how is your studies going right away? I want to discuss a few things about how are you revising, because we are going to finish a few units. Class 11 syllabus will be soon complete. So I wish to take a few things as well today. So first let me remind you that we had finished some parts of photosynthesis in the last class, right? So let me begin from where I left off. Alright. So we have completed light reaction. Alright. What is included in light reaction? All the reactions that are used to make two things: ATP, NADPH2, and oxygen is a waste product. These two things together will be used in the dark reaction to make glucose. And what are the steps involved in here? We have to take basically carbon dioxide, and there is a 5-carbon compound called RUBP. It combines with this carbon dioxide, it makes something, and this 6-carbon compound decomposes. So we get 2 into 3 molecules of phosphoglyceric acid. This is converted with the help of ATP and NADPH2 into a thing called triose phosphate. Actually it is 3-phosphoglyceraldehyde, to tell you the reality. Or the other compound is dihydroxyacetone phosphate, but no matter. So basically this is a 3-carbon compound, and we get, because we are getting 6 carbons, so here is also 6 carbon, here also we have 6 carbon. Now we regenerate RUBP, regeneration of RUBP. This is carboxylation, and this is reduction. So these are the 3 basic steps of dark reaction or light independent phase, and from this you extract 1 carbon which is in reduced form. So let us say it is in CHOH form. So if you run this cycle called Calvin cycle or C3 cycle 6 times, you will get 6 carbons which are reduced, right? So 6 carbon which are reduced, this equals to glucose, right? Okay. So this is a summary of what we have studied in the last class. Okay, I hope you remember all this. Today we will cover a few parts of the light reaction as well as the dark, the C3 reaction and something else called the dark reaction. Right, there is some. Okay. There is some issue, I believe, with that. Okay, there was a file I wanted to show you. No problem. So let's move on. We have completed light reaction, we have completed dark reaction. Now what remains is we study about photorespiration, we study about C4 cycle, and we study about CAM plants, right? So once again, what is light reaction? Light reaction is a process in which you make energy, you make reducing power, and you make oxygen. Light reaction is divided into two phases. One is called cyclic phosphorylation, other is called non-cyclic phosphorylation. So what is the difference between cyclic and non-cyclic? What is the difference between cyclic and non-cyclic? Do you remember? I told you the difference between cyclic and non-cyclic. You can also refer to your notes if you do not remember. I'll give you a minute to find out the differences. If you cannot do it, I'll tell you all of that by myself. Anybody? Okay, I understand it's cold and nobody wants to get up from their seats, but you can maybe try. Yeah. It's cold, I understand. Everybody is trying to keep their hands in their pockets, and very few people have just been putting both the hands on the table, right? Okay, so I can see. Alright, so it's okay. So you're warming your hands and all that. Okay, so if you're not telling me, let me tell you again. Look, light reaction is of basically a process in which you utilize light energy to make suitable compounds for dark reaction. And there are two things that you make which are very important, and that is ATP, and the other thing is NADPH2. But in cyclic photophosphorylation, you make only and only ATP. In non-cyclic, you make ATP, NADPH2, and the third thing, oxygen also. NADPH2, the reducing power, and oxygen is not produced in cyclic photophosphorylation. Also in non-cyclic, you make use of photosystem 2 as well and photosystem 1. And in cyclic, which photosystem do you use in cyclic? Only PS1. Only PS1 is used. So after that, we studied dark reaction. Dark reaction was studied by Melvin Kelvin, and he called it basically the C3 reaction. He gave the name because the first product, the first stable product of C3 reaction is a 3-carbon compound called 3-phosphoglyceric acid or 3-phosphoglycerate. It's a 3-carbon compound. 2 molecules are formed from one reaction. Yeah, okay, good. So finally you answered, yeah, it's PS1 in cyclic photophosphorylation, right? And now we are going to take all these things, put them together, and then we are going to make something called the first product, the C3 product. From this C3 product, you are going to make another C3 product, 3-carbon product called triose phosphate. From triose phosphate, 2 molecules, you are going to extract 1 carbon out of them, and from this 1 carbon, what are you going to do? From this 1 carbon, you are going to take this carbon, and this is the reduced carbon, and 6 times the cycle will be run, and after that you can get glucose. This part should be clear to everybody. And the enzyme that is responsible for making C3 compound, it is called Rubisco. I have told you that it is the most abundant protein in the world. It is the most abundant enzyme in the world, right? 50% of the all protein found in the world, this earth, is Rubisco. Okay. Full name of Rubisco is ribulose biphosphate carboxylase oxygenase. Alright. So all these things are clear. So this is C3 reaction. C3 reaction is basically easy. You have only 3 steps. What do you make out of it? It's glucose. Now I have to tell you one more thing about the cost of producing one molecule of glucose. The cost of producing one molecule of glucose. Does anybody remember what is the cost of producing one molecule of glucose? What is the cost? It's 18 ADP, ATP and 12 NADPH2. To make one molecule of glucose, we require 6 molecules of carbon dioxide. That is already you know, but the energy requirement and the reducing power requirement is this much. And how do you make 18 ATP and 12 NADPH2? In one cycle of non-cyclic photophosphorylation, you have been making 2 ATP, 2 NADPH2. Okay. So can you calculate how many times will you have to run cyclic and non-cyclic to make this much product? Can you tell me? To make 18 ATP and 12 NADPH2, how many times will you run cyclic and non-cyclic? How many times will you run cyclic and non-cyclic photophosphorylation? That's what I want to know. So can you tell me? No idea? 6 times, right? 6 times cyclic and 6 times non-cyclic. 6 times cyclic phosphorylation and 6 times non-cyclic photophosphorylation. Okay. All right. So this part is clear to everybody. Now please put the heading C4 plants. C4 plants and C4 cycle. Okay. So can you tell me what are C4 plants? What are C4 plants? Can you tell me? Anybody? What are the C4 plants? Nobody? Okay. So I'll tell you a story how this happened. Scientists were studying basically that there are some plants which produce fruits like mango. There is a mango tree and it produces very sweet fruits, but it takes a lot of time. Okay, it will take about 4-5 years for it to grow, and after that it will start producing its fruits. But these fruits are taking a large amount of time, right? So next the question becomes that after you do this, after the fruits have been made, can we calculate how much energy is required in producing these things? How much energy is required in producing the photosynthetic end product, the sugars, the basically the other forms of sugar, basically carbohydrates, right? So when this was being calculated, it was found that some plants are extremely efficient compared to some other plants. Okay, and these plants were found to be sugarcane. Sugarcane is at the top of photosynthesis, It is the most photosynthetically efficient plant on earth. That is why sugar is being produced in such huge quantity from a sugarcane plant, right? And so sugarcane was at the top. Then we have maize plant, we have sorghum, barley, right? So these plants are found to be more photosynthetically efficient compared to most of the plants. So the reason was being studied why these plants are more efficient. So some scientists found out that these plants have a special mechanism of photosynthesis and they have a different anatomy of leaf compared to the other plants. This anatomy is called Kranz anatomy. Okay, I'll write this down, all of these things. Kranz anatomy is a type of anatomy when you have a wreath-like structure, a crown-shaped structure. So it was found that they have two types of cells, and these type of cells are differentiated on the basis of anatomy, their position, and the type of chloroplasts. Yes, there are two types of chloroplasts found in sugarcane, and they have a special type of anatomy called Kranz anatomy. So when this was studied further, it was found that there is a very big important difference between other plants and these plants, and that difference is that when you study the first carbon dioxide fixed product, it is a 4-carbon compound, not a 3-carbon compound. So that is why these plants were called C4 plants. So when this difference became clear, the cycle was studied in great detail. It was found that they have a 4-carbon first photosynthetic carbon fixed product is called a 4-carbon product. Okay, so I'll discuss this in time. Now the question becomes that they are producing something else except of beside the other plants. So what is the use of this cycle? Why make something like this? So do you know the answer why these plants make a 4-carbon compound instead of a 3-carbon compound? Do you know why they do this? There must be a reason, right? Plants always have a reason. So can you tell me what is the reason? It occurs in mesophyll. Yeah, exactly. Kranz anatomy is found in mesophyll. The reason is C3 plants have the habit of making, eating and wasting. They waste a lot of glucose, and this glucose wasting process is called photorespiration. Okay, photorespiration I'll deal with later. So photorespiration is a wasteful process in which plants waste a lot of their fixed carbon or waste a lot of carbon dioxide. So because of this process, they lose a lot of energy. A lot of energy is not being used to make glucose, it is used to make something else, which releases carbon dioxide instead of fixing it. Because of this process, C4 plants become more efficient because they make no use of the oxygenic property of Rubisco enzyme. Yes, in C3 plants, Rubisco enzyme is responsible for carboxylation, and it is also responsible for the wasting process called the photorespiration. The full name of Rubisco is ribulose biphosphate carboxylase oxygenase. It can also accept oxygen instead of carbon dioxide in conditions of high temperature, high sunlight, low carbon dioxide and high oxygen saturation. So this process will likely happen. In C4 plants, almost zero photorespiration is found. Why? Because there is a concentration difference in these plants that is being maintained by forming a 4-carbon compound. They make a lot of 4-carbon compound and then pump it inside the cells where Rubisco is present. Where Rubisco is present, these plants, they pump the carbon dioxide in form of a 4-carbon compound and they artificially increase carbon dioxide concentration so much that no photorespiration will be happening. And because of this, there will be a slightly less, more requirement of ATP. They use more ATP to make glucose, still they become more efficient than C3 plants. The cost of making glucose in C4 plants is 30 ATP, 12 NADPH2. And now please see this here. I have just told you that in C3 plants, let's write, in C3 plants is 18 ATP and 12 NADPH2. So you can see the difference, right? 30 ATP. C4 plants will be using more ATP, same NADPH2, but more ATP. Still they are more efficient. So why is that? Because C3 plants have a habit of wasting so much. So that is what's exactly happening in our society as well. The students who do not waste a lot of time, they always win in the end. So please keep your focus on the exam. So I know this is a funny way of saying it, but still, please keep your focus on the exam, and I'm sure you will win, right? Okay, so let's continue. There are some plants in which it was discovered that the first product of CO2 fixation is a 4-carbon compound and not a 3-carbon compound. So these plants were called C4 plants. It was also discovered that these plants are more photosynthetically efficient compared to C3 plants. C3 plants in which the first carbon compound is 3-carbon, right? So it was also discovered that these plants are more photosynthetically efficient compared to most plants. And they have a different type of anatomy present in the leaves called Kranz anatomy. So please write this down quickly. I hope this is not blocking it. So yeah, let me know if you want me to scroll. Okay, so let's continue. These plants also have a different anatomy, different type of anatomy or construction, anatomy called Kranz anatomy. Kranz anatomy. And in Cran's anatomy, you find that there are two types of chloroplasts. There are two types of chloroplasts found in these plants. Two types of chloroplasts found in these plants. And we have to discuss this today. We have to discuss this, right? What are these different types of chloroplasts that we are talking about? Okay. Yeah, exactly. As you answered that it occurs in mesophyll, we have to consider this now, that what is this and how it is happening. So let me, I hope you have written it down, right? So what is this Cran's anatomy? So please write it down. There were two scientists called Hatch and Slack, and they discovered it. There were two scientists called Hatch and Slack, and they discovered it. So basically, Hatch and Slack studied this pathway. Wait a second. Hatch and Slack can come later. Let me finish something else. These plants are adapted to hot and dry climate. In continuation, please write down after this. These plants are adapted to hot and dry climates. Hot and dry climates. Okay. And there are the two types of chloroplast. The dimorphic chloroplast are basically found in the mesophyll, having two types of chloroplast. They are also called having two types of chloroplast. Is called dimorphic chloroplast. Dimorphic chloroplast habit. Dimorphic chloroplasts. So first is the chloroplast of the mesophyll cell, the other is the chloroplast of the bundle sheath cell, and we will discuss this. So because you have written it down, so next you can write, okay, the chloroplasts. Now please note, this is an important point. Which chloroplast is larger, it has been asked. The chloroplast of bundle sheath cells are larger in size. What is the difference? You must remember this, because it comes very frequently in true and false statements, and it becomes very confusing. He will say the chloroplast of choose the correct statements out of the following, and the statements can be the chloroplast of mesophyll cell is larger than the bundle sheath cell. So that becomes very confusing if you do not know exactly which is larger. So at present the question pattern of CBSE NEET exam is such that he wants you to pick a lot of true and false statements and count them. At 4 or 5 statements are given, count the correct number of true statements out of the following. So in that you have to analyze a lot of data like this. The chloroplasts of bundle sheath cells are larger than the chloroplasts of mesophyll cells. than the chloroplasts of mesophyll cells, right? Okay. Also, the chloroplasts of mesophyll cells, the bundle sheath cells are arranged centripetally and arranged centripetally. That means towards the center. Centripetally. So centripetal arrangement or larger size arrangement, this is important. Now the next statement is also very important, so I am making it with a different color. This statement has also been asked. It was asked in board examination also, so I am expecting it can come any time in the NEET exam as well. So this is, they contain starch grains. I am adding by myself because this is something else. They contain starch grains but do not have grana. Grana are absent. This is called agranal. Grana are basically absent. This is an important thing. Agranal. Okay, so please remember it. So if you talk about the chloroplasts of mesophyll cell, they will have less starch or no starch, and they possess grana. So next statement becomes the point of difference between the bundle sheath cells and the mesophyll cells. So let's take it down. This is down quickly. The mesophyll cells. On the other hand, have normal chloroplasts. What does this mean? Their chloroplast, whatever structure you have seen, grana, starch, okay, all those things will be present. Okay. So what is the only major point of difference between mesophyll and bundle sheath cell chloroplast? The mesophyll cells are normal type of chloroplast, that means they contain grana, but the bundle sheath cells, they will not have this kind of thing. Okay. So let me now draw the diagram of this structure. I hope you have written it down. So let me draw the diagram now. So this is the epidermis. This is the epidermis. This is going to be the upper epidermis. Right. Then we have the lower epidermis as well. So between them I have to draw a lot of structures here, and the lower epidermis you will also have to show stomata. So I'll draw this stomata in here somewhere. Like this. So this is suppose the stomata with the guard cells and all that. Now in the center I am going to draw these type of cells like this. Yeah, you want me to scroll up, I think. Okay, all right, it's okay. So these are the inner type of cells. So bundle sheath cells are these kind of cells, okay, and you will draw them like this. These are the bundle sheath cells, and in between them you can draw a little bit of xylem and phloem. So xylem, I am drawing it like this. So this is just present outside the vascular bundle. Xylem we always just draw normally with blue color. That has been by convention that I draw phloem with red color, but when I have to draw xylem, I normally use blue color only. So this is phloem, and outside all of this is the bundle sheath cell. Now in this bundle sheath cell, you will find a lot of chloroplasts, and they are like this. These are the chloroplasts. They are all looking towards the center. So this is called the centripetal arrangement of chloroplast, like this. Clear? This should do. Okay. So these are the normal. Sorry, these are the less. Okay. Now let me draw the other type of cells, which are the mesophyll cells. So it has not been so light as I wanted. Slightly light and between that. This should do. Actually want to draw the mesophyll cells now. So yeah, okay. The mesophyll cells, I am drawing it with somewhat lighter color, like this. These are the mesophyll cells, and then I will also draw the chloroplast inside these cells. Okay. I have to draw the cycle after this also, C4 cycle, and then I will draw something else also here. These are basically very large cells. Mesophyll cells are very large cells. Like this. Okay. Going down. Like this. These are the mesophyll cells. Now I must draw the chloroplast inside these cells. Let me choose something larger. So basically I have to draw a lot of chloroplasts in this. So these are normal random chloroplasts, nothing special about them. no special centripetal or centrifugal arrangement, just like they are present in a normal cell, these are present here. Right. So this diagram is complete. Let's do the labeling part. This is the upper epidermis. These are the mesophyll cells. Mesophyll cells. Then there are the chloroplasts of mesophyll cells. Then we have xylem, phloem and all that. These are the bundle sheath cells. This is the phloem. This is the xylem. Right. This is stoma, plural is stomata. Lower epidermis. Lower epidermis. Okay. Now these chloroplasts of bundle sheath cells, they have these chloroplasts agranal or without grana, agranal chloroplasts of bundle sheath cells. Now this diagram is complete. So let me know as soon as you have finished. Okay. I have to make one more diagram after this. That is going to be an equation type of diagram. Nothing else about that. Alright, you have finished, I guess. Okay, good. So Shillong you have copied, Itanagar and Tamenglong. If somebody else remains, let me know. So carrying on, now let us discuss the reactions of C4 cycle. What is happening here? The C4 cycle, the C4 cycle or the Hatch-Slack pathway. Let us start it. Hatch-Slack pathway reactions. So we are going to draw two cells. First is the mesophyll cell. So let us choose again. For that we will take a this type of cell we will take. Okay. Let us take something else to fill it with. This is a mesophyll cell that I am drawing. I am just labeling it. Give me a second. be done. This is a mesophyll cell basically. This is a mesophyll cell. Just adjacent to the cell, we will place another cell and we will call it the bundle sheath cell. So, what will be the chapter for this Saturday test? For botany, you say? Give me some time, I will let you know. Okay, I will let you know. Okay, no problem. I will just let you know. This is another type of cell, that is the bundle sheath cell. We have to write a lot of reactions here. Just give me a minute. Right. Now we can begin what we wanted to do originally. This is a bundle sheath cell. Very few reactions are taking place in bundle sheath cell. Basically, what I have to write, that is going to be very short. Otherwise, the whole C3 cycle is happening here only. So, the mesophyll cell, what happens? Carbon dioxide is entering from the outside into the mesophyll cell. The mesophyll cell is taking in carbon dioxide from the atmosphere. So, by the stomata, the carbon dioxide is going to enter into the mesophyll cells, and this CO2, it happens to interact with phosphoenol pyruvic acid. PP stands for pyruvic acid, and phosphoenol pyruvic acid, it is a 3-carbon compound. It interacts or it reacts with phosphoenol pyruvic acid and carbon dioxide react together, and they produce a 4-carbon compound called oxaloacetic acid. I am writing the full name here because it is very important. It is a 4-carbon compound, oxaloacetic acid. This is the first product of C4 cycle. This is the first product of C4 cycle. Okay. And oxaloacetic acid, it undergoes a change by the help of NADPH2 NADPH2 gets converted into NADP plus, and it gets converted into something else called malic acid. Malic acid is what gets converted into malic acid. Malic acid is a 4-carbon compound as well, and this malic acid, it gets entry inside the bundle sheath cell, and here it gets decarboxylated. It gives out carbon dioxide. This carbon dioxide is now going to enter into the C4 pathway. Sorry, C3 pathway, called the C3 cycle or Kelvin cycle. So, Kelvin cycle, it will enter inside the Kelvin cycle with the help of this reaction. So, bundle sheath cell, it will again now have a 3 carbon compound called pyruvic acid. Pyruvic acid. It's a 3 carbon compound. Okay. And pyruvic acid, it gets transported back to the mesophyll cell. Pyruvic acid, it gets transported back into the mesophyll cell, and then the pyruvic acid gets converted into phosphoenol pyruvic acid. Phosphoenol pyruvic acid by the help of ATP. ATP gets converted into adenosine monophosphate plus inorganic phosphate. ATP always get converted into these products, if you remember correctly. ATP gives AMP plus inorganic phosphate is written as IP, small I capital P, and it gets converted into a 3 carbon compound again called phosphoenol pyruvic acid. Are you getting me? So, what are the changes happening here? Please look at it again. Carbon dioxide enters from the atmosphere. So, let me write here, atmospheric carbon dioxide enters the mesophyll cell, forms oxaloacetic acid. So, oxaloacetic acid is the first product of C4 cycle, or first photosynthetically fixed compound. It gets converted into another 4 carbon compound called malic acid. I have written the number of carbons on each and everything. There is one more thing that you must remember. When malic acid enters the bundle sheath cell and it gives off carbon dioxide, it can regenerate the NADPH2. NADP plus gives NADPH2. So, whatever NADPH2 was used, it is regained. It is regained. It is regained. Right? It's regained. So, finish it quickly, then we will move on further. Okay, so this is done. Finally, so you have done it. Okay, so let me come back, give you a quick recap what's happening here. The atmospheric carbon dioxide, it is going to enter from this side. CO2 is going to enter from stoma here. It is going to enter into the mesophyll cells, and after the mesophyll cells have made a 4 carbon compound, it is finally going to enter into the bundle sheath cell. So this is the pathway that it is going to take. Presence of two types of chloroplast is characteristic feature of C4 plants, right? So this is clear. CO2 is finally entering inside the bundle sheath cell. So the main reactions of photosynthesis, the dark reaction and everything, it is taking place inside the bundle sheath cell. In mesophyll cell, light reaction can happen. In mesophyll cell, a dark reaction does not happen. I hope you understand me. In mesophyll cell, they are basically transporting the maximum amount of carbon dioxide to the bundle sheath cell. So if they have to perform photosynthesis dark reaction, they can do that. It's no limitation. But they try to transport maximum carbon dioxide inside. So that is why there is not much dark reaction happening in mesophyll cell. However, some amount of a little bit of photorespiration can happen. But because bundle sheath is saturated with carbon dioxide, so therefore little or no, absolutely nothing of photorespiration will happen in bundle sheath cell. So that is what we had to do, the main reactions. So let's write the remaining part. Okay. The other names of C4 cycle are to be discussed now. The detailed study of C4 cycle was carried out by Hatch and Slack. So it is also called Hatch-Slack pathway. Hatch-Slack pathway. You have a question, right? Very good. I'm happy somebody asked me this question. Sir, why adenosine monophosphate and not ADP? Yeah, very good. So let me explain to you this thing. You will need about a minute to understand this. So on your page somewhere, find some space on the top of your margin somewhere. I want to tell you something. Look, this is adenine molecule. This is adenine molecule. Actually, it's a pentacyclic ring, but it's okay. Adenine molecule is attached to one phosphate. The first phosphate that the adenine molecule is attached to, this is phosphate group. This is a low energy bond. Low energy bond. Then this molecule is called adenosine monophosphate. Then we introduce another bond, another phosphate bond, the second phosphate bond. Now this molecule becomes adenosine diphosphate, and this bond is a high energy bond. The second bond is a high energy bond. If you break this bond, a lot of energy will be released. High energy bond. Then there is a third phosphate bond. It is also a high energy bond. Bond energy of second and third bond is equal. Now you call it ATP. Now you call it ATP. So adenosine triphosphate has 3 phosphate bonds, out of which we normally break the first bond only. Out of which we normally break the first bond only. So when we break the first bond only, we get adenosine diphosphate and one inorganic phosphate. Plus large amount of energy. Normally we do not break the second bond because it acts as a sort of energy reserve. Okay, but if you convert one ATP into AMP, you are consuming 2 moles of high energy phosphate bond. That is equivalent to 2 ATP. If you break in 2 bonds, it is equivalent to 2 ATP. Now students, let's come back here. Come back here. What is the energy requirement of C4 cycle? To make phosphoenol pyruvic acid, from pyruvic acid you are consuming 0 NADPH2, because NADPH2, one is consumed, one is liberated. You can see it here, right? But what about ATP? You are consuming actually 2 moles of ATP, or 1 mole of ATP converted into 1 mole of AMP. So that's an equal way of saying you are consuming 2 ATP per cycle of transport. Is that clear? You are consuming 2 ATP per cycle of transport. Now please remember, what is the cost of photosynthesis in C3 cycle? 18 ATP, 12 NADPH2. What will be the cost of photosynthesis in C4 cycle? Can you calculate? Total 6 molecules used to make 1 glucose. For 1 CO2 transportation in this cycle, you require energy equivalent to 2 moles of ATP, right? So what will be the total energy requirement? C3 plus 6 into 2, that is C3 plus 12 ATP. C3 is 18 ATP. So your net getting is 30 ATP and 12 NADPH2. Now you remember why I told you that this cycle is more expensive, yet it is more efficient. Remember that? In the beginning of the cycle I told you that this cycle is consuming 30 ATP. Where is this coming from? It is coming from here. I hope it is clear now. Do you want me to repeat, or is it clear? Do you want me to repeat, or is it clear? Should I explain again? You can consume 1 AMP. Okay, look. Here ATP is converted into AMP. So you breaking energy, either you do this, or you can also write 2 ATP. 2 ATP consumed into 2 ADP. To make 1 phosphoenol pyruvic acid, you need 2 ATP, or you need 1 ATP, and this process normally how it is happening, normally it is consuming ATP and it is liberating AMP. It happens by itself. We are not doing it on our own. It happens by itself. So if you are calculating the energy requirement of transporting 1 carbon dioxide molecule, so NADPH2 is consumed in making malic acid. But when malic acid is breaking down, it liberates 1 molecule NADPH2 again, right? So 1 plus 1 minus equal to 0, 0 NADPH2. And CO2, when it enters the C3 cycle, again 18 ATP, 12 NADPH2 will be used, just like Kelvin cycle. That is fixed. But what about the transportation cost? Transportation cost here is extra. How is it coming? It is coming from pyruvic acid and phosphoenol pyruvic acid. In that reaction, energy equivalent to 2 phosphate is consumed. So you will not write 1 ATP is consumed here. You can write, when you have to calculate the cost, then you will say that energy equal to 2 ATP is consumed in this total cycle, or in the reaction between formation of PP from pyruvic acid. Let me write the full name of PP, phosphoenol pyruvic acid. So to make PP, to make phosphoenol pyruvic acid, you need 2 ATP. You need actually 2 ATP, or you can also say 1 ATP making 1 AMP. That is equal. Why? Because the energy of second and third, this bond and this bond is equal. The energy of breaking, this is the first phosphate, this is the second phosphate, this is the third phosphate. That is why I showed you the structure of ATP. Okay, this bond is equal to this bond. This bond is equal to this bond. Second and third bond is equal. Second phosphate bond, breaking of that bond is equivalent to breaking of third phosphate bond. Same energy. So if you make, normally we always break ATP into ADP, but if in some cases AMP is produced, then you are consuming energy equal to 2 ATP, because normally we do not make AMP or we do not break AMP further. Okay, why do we do this? Because it's body's mechanism to save energy for very dangerous reactions or emergency reactions. Suppose somebody is not getting oxygen at all, then body needs energy to survive. Brain cells need some energy to survive. So at that time we will break the second bond and the first bond in very severe conditions. Normally we will never break it. Normally we will always convert ATP into ADP only. But if you do that in some reaction, then you are consuming more energy equal to 2 ATP. Are you able to understand it now? Okay, thanks. So finally you understand it. So if you are making AMP, you can also write 2 ATP, that is equivalent. You can also write, if you are getting confused, you can also write 2 ATP gives 2 ADP. End of story. I just wanted to tell you the exact thing what is happening, but if you are getting confused, you can also write this. Okay, it's normally not required, but if you are getting confused, you can write it like this. So this is done. So now let's write the energy expenditure in C4 cycle. Energy or cost of making one glucose from 6 carbon dioxide in C4 cycle is equal to 18 ATP plus 12 NADPH2. This is fixed. This is fixed. It is from C3 cycle only. It is from C3 cycle only. Plus 12 ATP. Because in transporting one carbon dioxide you need 2 ATP or 1 ATP into AMP, that is equal to 2 ATP only. So you have 6 carbon dioxide, so you need 6 into 2, 12 ATP. So total it comes to 30 ATP plus 12 NADPH2. ATP plus 12 NADPH2. This is the cost. Now let's write some examples. Also, there's something else also you need to write. C4 cycle is also called DCA cycle or dicarboxylic acid cycle, because oxaloacetic acid, that's a dicarboxylic acid. All right. So now you have copied, right? So please also write. It is also called DCA cycle or dicarboxylic acid cycle, because the first stable product is a dicarboxylic acid. So beside Hatch and Slack cycle, you also call it DCA cycle, you also call C4 cycle. So there are many names for this cycle. Okay. Now let us write the examples. Okay. And few things also. Sometimes a name of enzyme used in this reaction is also asked. So I will just write it down. So phosphoenol pyruvic acid, PP, converted into oxaloacetic acid. This reaction is equivalent to Rubisco reaction, ribulose biphosphate carboxylase. So we call it Rubisco, RUBP carboxylase. Similarly, the enzyme which is accepting the carbon dioxide here, it is a carboxylation reaction. So this enzyme which is used here, it is called PEP carboxylase. I have not written it here because of less space. In short, you can write it just like Pepsi or Pepco. Because of less space, I did not write it. Pepco is the name of enzyme. PPCO. Pepco. Okay. Pepco is the name of the enzyme. So a question has been asked in NEET examination. Among RUBP carboxylase and PP carboxylase, which enzyme has more affinity or more love towards carbon dioxide? I want you to guess the answer. You have Rubisco, you have Pepco. Which enzyme has more affinity towards carbon dioxide? I am sure you are solving NCERT fingertips, MTG fingertips or some other books as guide questions. So this question you will find in a lot of competitive books. Pepco and Rubisco, which enzyme has more carbon dioxide affinity? Pepco, yeah, exactly right. So please write it here because it has been asked in your examination. It's a very important thing. Actually, Pepco has 80 times more affinity than Rubisco. 80 times. But no, you don't need to write the exact figure. Pepco has more affinity for CO2 than Rubisco. It has more affinity for CO2 than Rubisco. Okay. All right. So final thing is, this is done. This is done. Now let us finally write down the examples. You can also remember, if something else is asked, what is the primary carbon dioxide acceptor molecule in C4 plants? What is the primary CO2 acceptor in C4 plants? Primary CO2 acceptor, you understand? The compound which first of all accepts carbon dioxide. So I think the answer is clear. It is PP. PP is the primary electron acceptor in C4 plants, and in C3 plants the primary electron acceptor is RuBP. There is a difference, right? Just please pay attention to these type of questions. So PP is the primary electron acceptor in C4 plants. Last part of this C4 cycle is examples of C4 plants. C4 plants, sorry. So what are the examples of C4 plants? One I have already told you, sugarcane. Maize is another example. Please write. Sugarcane name is Saccharum officinarum. Botanical name is Saccharum. Most common species is officinarum. Maize you already know, Zea maize. Sorghum vulgare, sorghum, amaranthus, right? Then you have cypress, then you have digitaria, out of these, this has been asked, this has been asked, this has been asked. Digitaria is normally not been asked, so I have put it in separate column. All these plants have been asked in the exams, so all these are important. Which of the following is an example of C4 plant? Question like this. Which of the following plants, indirect question can be asked, which of among the following plant, they will give 3 C3 plants and 1 C4 plant, which among the following plants has 2 types of chloroplast? Language of question can change. They will not ask you directly C4 plant. They will ask you indirectly, which of the following plants has 2 type of chloroplast. So sugarcane has, maize has, sorghum, amaranthus, cypress, all C4 plants, right? So this type of question can be asked. C4 cycle is finished. Let's write CAM plants or CAM cycle. Okay. So this is done. Let us write down CAM or crassulacean acid metabolism. I hope you have written it down. You should have scrolled to next page. Okay. Crassulacean acid metabolism. or CAM. So it's not a video camera. It's just because of some plants, they belong to a family called Crassulaceae, and on this Crassulaceae family it was first discovered. Okay. So this cycle is remarkably similar to the C4 cycle that you have just studied. But there is one important difference. These plants operate this mechanism only because they have a water deficiency conditions. They have a very strong water deficiency condition, and so these plants, they will never open their stomata during the daytime. They will open it only during the night time. So it's just like closing your nose for the rest of the day and breathing only at night. Okay, stomata is like the nasal pores of the plants. So the plants, what do they have their habit? They close their nose during the day, and during the night time they perform a lot of yoga, right? They perform a lot of exercises for intake and expiration and inspiration, and they take a lot of carbon dioxide inside and they store it in their cells. So that is what you have to do. So this is basically a two type of what happens during the day, what happens during the night. So let's write it down. It is a mechanism the CAM plants have developed a mechanism for CO2 fixation during the dark time, during night time, normally when is CO2 fixed? It is fixed in the daytime. But in these plants, the carbon dioxide is fixed during the night time. It operates in succulent plants. Succulent plants. Example, let's write a few examples. Cactus or opuntia, same thing. Then you have pineapple, then you have bryophyllum, Kalanchoe, sedum, agave, americana, the monocarpic plant which produces flowers in about 100 years. Bryophyllum is done. Pineapple you know already, and orchid. Orchids also show this habit. Okay. etc. Alright. So now I have to draw this cycle. This is what is happening during the night time. This cycle will have some part which is completed during the night time. Have to make some more space for it because it has a lot of reactions like this. So this phase is going to happen in the night time. So there are some stars here, a moon here as well. Okay, so guess you get the idea. And then you have a phase which is happening during the daytime. Alright, so this will do. You will be able to understand what's happening in day and what's happening in the night. Okay, so let me continue. So in the night time, what is happening? In the night time, phosphoenol pyruvic acid (PEP), phosphoenol pyruvic acid, it will accept the carbon dioxide. Okay. It will accept the carbon dioxide. The stomata are open. The stomata are open. Stomata are open. So carbon dioxide can enter inside the plants like this, and this will combine to make oxaloacetic acid. So here also the first product of carbon dioxide fixation is oxaloacetic acid. It's a 4-carbon compound. It's a 3-carbon compound, and carbon dioxide you already know it's a single carbon compound. Now this oxaloacetic acid, it will be converted into malic acid. It will be converted into malic acid. Malic acid. And what will be consumed here? You will consume NADPH2 to make NADP plus, and you will reduce oxaloacetic acid into malic acid. Okay, and this malic acid, it will be transported, it will be stored in the cell vacuole, and this will be utilized during the daytime. So, I am writing malic acid here in that same cell where malic acid is formed. In that very same cell, it is not being transported. It is not being transported. It is not being transported, right? So, in that very same cell, this malic acid is now being converted by decarboxylation and by reducing NADP plus into NADPH2 again, like this. Like this, malic acid is converted into pyruvic acid. It is converted into pyruvic acid, and carbon dioxide is being released. This pyruvic acid, it will then, you know, the how it will be converted into phosphoenol pyruvic acid. So, you can write here, at the daytime the stomata here are closed, stomata. The gate is closed. Stomata is closed, and then this pyruvic acid, it will be converted into phosphoenol pyruvic acid in the daytime, in the okay. So, how will it happen? You know, it will happen by the help of ATP being converted into AMP, right. So, this is what is happening, and this carbon dioxide, it will be accepted by RUBP, ribulose biphosphate, and it will be run in the Calvin cycle, C3 cycle, Calvin cycle, and what will come out of Calvin cycle? The carbohydrates. Like this. I hope you have copied it. Let me know when you are finished. Let me know when you are finished, right? Okay. All right. So, this is done. Finally, so we have to write some few things about CAMP cycle also. In order to conserve water, these plants have some special adaptations. order to conserve water, CAM plants have some spatial adaptations. What are these adaptations? They open their stomata only during the night time. This is called scotoactive stomata to conserve water. Because at the night time humidity increases, temperature decreases, so there will be little or no loss of water by transpiration. Okay, there will be no transpiration. Actually, you can also write this is called scotoactive stomata. Scotoactive stomata. Okay. The carbon dioxide, they store carbon dioxide in night time. In the form of malic acid. In the form of malic acid. In presence of enzyme PP carboxylase. PP carboxylase. Okay. Next, you can write the CO2 stored during night is used in Kelvin cycle during daytime. Okay. Also, at the daytime, whatever respiration is happening, that is also used in photosynthesis. Okay. Also, whatever carbon dioxide is released in respiration is consumed in photosynthesis. This is happening during daytime. Is consumed in photosynthesis. Is consumed in photosynthesis. Now there is one more interesting phenomenon that I want you to remember. In the night time, a lot of carbon dioxide is being stored. So can you tell me that if you take opuntia during let's say in the evening and in the morning, at which time will you find that it is more sour in taste? If you take cactus and you break a piece of cactus and remove the thorns from it, then try to eat it, at which time will you find it is more sour in taste? In the evening or in the morning? Okay, you do not want to eat alright, let me put it this way. You take a pineapple, okay, you take a pineapple leaf tissue, no, not the fruit, okay, fruit is normally will take taste sweet, and you take the pineapple leaf and you try to taste it. At which time will it appear more sour in taste? In the morning. Why? Because in the whole night time period it has been storing a lot of carbon dioxide. A lot of carbon dioxide is being stored, the pH is going to drop down drastically. pH is going to become very less. And all acidic things are sour in taste. Interesting question, I hope you enjoyed it. All sour things are acidic. All acidic things are sour in taste. All alkaline things are bitter in taste. Have you ever tasted soap? Have you ever tasted soap, anybody? By accident, I guess, right? So, all things that are alkaline, they will taste bitter. And all things that are acidic, they will taste sour in taste. Okay. So please write, there is decrease in pH. There is decrease in pH during night. In the night time, there is decrease in pH. So in the early morning, when photosynthesis is not started also, you will see there is a large decrease in pH during the night and increase in pH during the day. And increase in pH during the day. So in the same cell, look, in plants like C4 plants, C3 cycle and C4 cycle, they were both happening in different cells. C4 cycle was happening in the mesophyll cell and the C3 cycle was happening in the bundle sheath cell. Here it is happening in the same cell. In the same cell, both C3 and C4 cycles takes place. In the same cell. So this habit of these C4 plants, CAM plants, enables them to survive in very adverse desert conditions. Okay. This capability enables them to survive in xerophytic conditions. This capability enables them to survive in xerophytic conditions. Xerophytic habitats, right? The malic acid is stored in the cell vacuole. The malic acid formed is stored in cell vacuoles during night. Okay, so this is about CAM plants. Another interesting thing related to a C4 plant, pineapple. Everybody likes pineapples? Yes? Do you like eating sour pineapples or sweet pineapples? Do you want to eat the pineapples which are sweet in taste, or you want to eat pineapples which are sour in taste? I have a interesting experiment for you. Do you like sour or sweet? Which pineapples do you like? Some are very sour in taste and some are very sweet in taste. I can tell you something which can help you convert sour pineapples into sweet pineapples. And that will take only 5 seconds. Everybody likes pineapples, right? Pineapple is a CAM plant. Alright. No answer? Okay. Yeah, so what you can do is, I will tell you, take some salt before eating the pineapple, and take this salt and spray it on the pineapple. When you are eating the pineapple, before eating it, spray some salt on it and spread it evenly on all the surface. Do not use very less salt, you can get it with experience. Even if it is a sour pineapple and it is very sour in taste, it's very acidic, it's very sour. If you spray some salt on it and then you try to eat it, it will suddenly start tasting sweet. You can try it. Okay, this trick is used by fruit vendors to make their pineapples taste sweeter. When you cut them and go home and cut them, you will see that it's not so sweet when it was put on the display. So this is the trick that they use. Okay. And regarding pineapples and all these C4 plants, CAM plants, the thing is, pineapples take about 2 years to grow. It's a long time, 2 years. But the thing is, it does not require a lot of care. You can put it anywhere, do not care for it. After 2 years, you can go and cut the crop, because it has this CAM plant capability, so it can save a lot of water for itself. It will perform photosynthesis in the day by using the carbon dioxide taken in during the night. So we do not require to care for these plants a lot. They can survive by themselves. Alright? Yeah, sir, it's time. It's not time. Okay. You still have some minutes, and about 5. Yeah, I have some about 5-10 minutes. So let us try what we can do. Do not ask me. Okay, all right. So I'll give you the break now. All right. I guess because this is over now. Okay, so let's take. We'll do this in the next class. We'll complete photo respiration. And we'll also complete the Blackman's law of limiting factors. That is the last thing that is remaining in this chapter. Okay, so we'll do that tomorrow. Okay, thank you everyone. Thank you for your time. We'll do this tomorrow.